Hello, very good evening to the student who have joined. Please wait for some time. We will be starting our session soon. Okay. I hope my voice is audible and you can see my video and also the presentation. If not, please let me know through the chat box. We will start at 6 p.m. Till then, kindly wait. Yeah, very good evening. Okay, so thank you for confirming. Good evening to all the students who have joined. Please wait for some time. We will be starting our session at 6 p.m. Okay, meanwhile, let me know if there is any issue with the audio or with the presentation. I hope my presentation is visible and I'm audible too. We will be starting our session at 6 p.m. So, if anybody has any doubt regarding our last class or anything, you can let me know now. Okay. Okay, just wait for 2 to 3 minutes. We will be starting at dot 6 p.m. Okay, let few more students join, then we will be starting our class. Okay, so I think I will be starting with the introductions. Okay, so it's 6 p.m. So let me start the class by introducing myself. I hope my voice is audible and the screen is visible to all. If not, please let me know through the chat box. Okay, so very good evening to the students who have joined. My name is Mousumi Deb and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL SWAM course of Medicinal Chemistry, and this is our week 3 live session. So before starting the session, I would like to clarify a few things. like it is a weekly problem solving session. Okay, and the sessions are on Monday from 6 to 8 p.m. It is a 12-week course. This medicinal chemistry, you have a 12-week I mean course content. Okay, so we will be having 12 problem solving sessions each week, subsequently on Mondays from 6 p.m. And after ending this 12-week course, we will be having another class, which is a summary class to summarize the entire course. Okay, so all the classes are live on Google Meet platform, and this is the link for joining the class. This link will remain same for the Monday class for our whole semester. Okay, you have to use the same link to join the Monday classes. So if any network disruption happen from my side, do not leave the session immediately. I will try to rejoin as soon as possible. And since the session is open, you can rejoin at any time if there is any network issue or anything. You do not have to worry because I'm recording the session. Okay, so one more thing, that I have muted your microphones and cameras so that I can record the session without any problem. But the chat box is always open, and I will be communicating through you to you throughout the session via the chat box. Okay, so if there is any doubt after the class, you can drop an email to me, or throughout the session you can post your doubts and queries through the chat box. Okay, I will keep remaining. I will keep checking the this particular chat box to see if there is any doubt or query in class. If it is possible, if the time permit, I will try to answer your queries. And if your query I cannot answer it in the class, you can drop an email to me by asking your question. I will be answering in the query also via email. I will share my email ID at the end of the session. So as I have mentioned, I am recording the session, so the class recording and this presentation which I am showing today, it will be shared to you. Okay, so where you can find this class recording, and also where you can find this particular presentation. So I have shown you earlier also, but let me show you one more time. Okay, so you have to go to the SWAM portal. Okay, this is the announcement page of the medicinal chemistry course. I'm sure everybody who have registered for the course can easily log into this page. Okay, so in the left panel, you will be seeing there is a, when there is a weekly, I mean course content, I mean there is a release, there is a, at the end there is a problem solving session. So you have to click on that part, and you have to click on the problem solving session recording. Once you click on that particular I mean the link, you will be redirected to a page which looks like this. Okay, here you will be finding the class wise I mean YouTube link for the course recording, and also there we will be finding a particular Google Drive which contains the PPTs which I am showing in the class. Okay, so from here you can see all the things. Okay, you can check this later. So generally I try to upload the class material and also the PPT, or and also the videos at the in that particular night. Okay, and so you can check by tonight or tomorrow morning, you will be surely getting the class recording and also the presentation. Okay, so today it is our week 3 live session, as I have mentioned. So in our week 1, what we have learned, we have learned the various aspects and the introduction of medicinal chemistry. What is medicinal chemistry, how it works. Okay, so we have discussed the basic of that, and along with we have discussed about the basic structures of proteins, a little bit about amino acids, acid base, that concepts. In our second class, we have discussed mostly on enzymes, how the enzymes work, what is the enzyme kinetics, what are the things about them. Some mathematical questions also we have solved, and we have discussed about receptors, the type of receptors and the details that we have discussed in our week 2 course content. So what is in our week 3 course content? Let us see. We have the nuclear, regarding we will be discussing about the nucleic acids. Okay, we will be discussing about RNA and protein synthesis. Then we will be discussing about basics of molecular biology. Okay, like this protein synthesis and enzymes which are responsible for the same. Then we will be discussing on the enzymes as drug targets, enzyme kinetics and inhibition. Today vastly we will be discussing about the enzyme inhibition, because it is a very important topic, but previously we do not have discussed this particular enzyme inhibition. So so how we will be discussing regarding this enzyme, different modes of I mean inhibition, their kinetics, and we will be solving a few mathematical questions also. Okay, so let us start the session. So let me go to the first. So how we do it. So basically, I will be showing you around 20 MCQ questions. Okay, each question will be having four different options. One of which will be the correct answer. So after showing you a particular question, I will wait for 2 to 3 minutes. Okay, and you have to answer in through the chat box like A, B, C, or D. Which of the following will be the correct answer for a particular question? After 2 to 3 minutes, I will check the chat box. Okay, and then I will be showing you the correct answer of that particular question. Okay, so that is how we do in our class. So let me go to the first question of today's session. So the first question, the problem number one, it says that which of the following statements is incorrect? Your options are number A, the distance between two major groups of DNA is 34 Armstrong. Number B, the distance between two major groups of DNA is 10 Armstrong. Number C, uracil is not present in DNA but is present in RNA. And number D, linkage of the nucleosides happens through the phosphate group. Which of the following statement is incorrect? I will be waiting for a minute or two, then I will check the chat box. Okay. So whenever there is a correct, incorrect, true, false, that type of question, please read the options very carefully. Okay. Because sometimes this kind of question can be a little tricky or can be a little confusing. So read the options very carefully, then let me know in the chat box A, B, C, or D which of the following you think is the correct answer for problem number one. Which of the following statements is incorrect? Number A, the distance between two major groups of DNA is 34 Armstrong. Number B, the distance between two major groups of DNA is 10 Armstrong. Number C, uracil is not present in DNA but is present in RNA. And number D, the linkage of the nucleosides happen through the phosphate group. Let me know A, B, C, or D which of the following will be the correct answer. Let me check the chat box. Okay, regarding this issue, this class material of your Saturday class, I think you should put a email, okay, to the NPTEL. In the discussion forum, you can put your query like you will be finding there is a question answer discussion forum here. Okay, here you can drop your query or you can directly email. Okay, you here by adding new conversation, you can add up your query about the class material. Also, you can directly email to the SWAM portal, SWAM website, or to the authorities regarding that. Because Saturday class, I am not taking that particular class, so I do not know about this particular problem. Okay, I cannot help you regarding that. Or in the Saturday class itself, you can tell the particular concerned person who is taking the class regarding this issue. Only he or she can fix this problem. Okay. Okay, so I have only got one answer regarding the first question. See, in the for answering the class, since there is no negative marking or no that kind of issue, I mean, expect that all of you will try to answer the questions, even if your answer is wrong. Okay, another answer I have got, and it is also saying that it is number B. Okay, let me check if number B is the correct answer for problem number one or not. So the correct answer will be yes, number B. The distance between two major groups of DNA is 10 Armstrong. This statement is incorrect. Okay. Now let us understand what is the basic structures of the DNA and RNA and all these things. Okay. So basically, we all know that DNA and RNA both are the your nucleic acids, right? So the DNA, the deoxyribose nucleic acid, and RNA is the ribonucleic acid. So what is the difference between the DNA and RNA? First, let us understand what is the structure. So as you can see here, we have this is known as the nucleotide. Okay, so nucleotide is the this all nucleosides. Basically, first we have this nucleoside. What is the nucleoside? Is a base and the pentose sugar. So there are three parts of a particular nucleic acid. One is the nitrogenous base, then we have a pentose sugar, and then we have this phosphate. So what is this bases? So we have five type of base in case of the nucleic acid. We have this pyrimidines and purine bases. Okay, so purine base are this I mean this fused ring, fused of one six member and one five member ring fused to each other. It is known as a purine, and pyrimidine is a particular six member rings. Okay, so purine base we have this adenine and guanine. Pyrimidine base we have cytosine, thymine, and uracil. In DNA we have this adenine, guanine, cytosine, and thymine. Whereas in the RNA, in place of thymine we have the uracil, right? So this base is attached here in this 1' position of this pentose sugar, and this in this 2' position we have either OH or H. Whenever there is a OH, it is known as the ribose sugar, and when there is a hydrogen, this is known as deoxyribose sugar. Ribose sugar is present in RNA and deoxyribose is present in your DNA. So this base and this sugar together, it is known as the nucleoside. Okay, when there is a phosphate attached on the 5' portion, it is known as the nucleoside monophosphate. And then when two phosphates are present, it is known as the nucleoside diphosphate. And when three phosphates are present, it is known as the nucleoside triphosphate, or it is also known as the nucleotide. Okay. So these nucleosides, these are joined with each other. Two nucleosides are joined with each other with the help of this phosphate linkage. Okay. So Watson and Crick solve the secondary structure of DNA. So in like we have in our protein, we have secondary structure of protein, tertiary structure of protein, and also the quaternary structure of protein. In case of DNA and RNA, the things are same. Okay. So Watson and Crick model, we know about the DNA double, which has a double helix, which looks like this. Okay, Watson and Crick solved the secondary structure of DNA by building a model that fitted all the known experimental results. The structure consists two DNA chains arranged together in a double helix for the constant diameter. The double helix has a major group and a minor group, which are some importance to the action of several anti-cancer agents acting as the intercalators. Okay, the structure relies crucially on the pairing up of nucleic acid bases between two side chains. Adenine pairs up with thymine via 2 hydrogen bond, whereas the guanine pairs up with cytosine via 3 hydrogen bond. Thus, the bicyclic purine base always linked with a smaller monocyclic pyrimidine base to allow the constant diameter and double helix. The double helix further stabilized by the fact that the base pairs are stacked one up to each other, allowing the hydrophobic interaction between the faces of the heterocyclic rings. The polar sugar phosphate backbone is placed outside the structure and can form favorable polar interaction with the water. So as you can see, this double helix structure, so this is the different distance between one major and one minor groove. This diameter, it is around 10 angstrom, but the distance between two major grooves is the 34 angstrom. This so as you can see, if you number, this thing is number A, so as you can see, this is number 1 and this is the number 2. Okay, so between the distance between two major grooves is 34 angstrom, and distance between two the distance between the minor grooves are the 10 angstrom. So these are these things which we have to understand. And one more thing, these are, I mean, I hope, I know you have heard a lot, but this is the few very crucial things to remember, that there are this number of hydrogen bonds. Okay, so in case of DNA, adenine pairs up with thymine. In case of RNA, adenine pairs up with uracil, but the number of hydrogen bonds will be same, which is 2. Okay, and the cytosine and the uracil, guanine and cytosine is constant in I mean in both DNA and RNA. So number between the hydrogen bonds between the guanine and cytosine is 3. So as you can see, it is also shown in the structure. And this sugar phosphate backbone, basically it stays outside this helix, which is the reason of the favorable interaction with this particular helix with the water. Okay. So this is the structure of double helix, and this is all about the you have to remember. I know it may seem a little tough, but try to remember the structures of this adenine, guanine, and this cytosine, thymine, and uracil structures. Okay, this nitrogenous bases structure. It is better if you can remember the structures. Okay. Now let me go to the next problem. Problem number 2 says, what role does the messenger RNA play in the synthesis of proteins? Your options are: number A, it catalyzes the complete process; number B, it provides the genetic blueprint for the protein; number C, it creates the genetic code to the specific amino acid; and number D, it modifies the messenger DNA molecule prior to the protein synthesis. What role does messenger RNA play in the synthesis of proteins? Number A, it catalyzes the complete process. Number B, it provides the genetic blueprint for the protein. Number C, it creates the genetic code to a specific amino acid. And number D, it modifies the messenger DNA molecules prior to the protein synthesis. Let me know A, B, C or D. Which of the following will be the correct answer for problem number 2? Which of the following is the correct role played by the messenger RNA in the protein synthesis? Again, I will be waiting for a minute or two, then I will check the chat box. Try to answer the question. Again, I am saying even if your answer is wrong, it's completely fine. Okay. What role does the messenger RNA plays in the synthesis of protein? Number A, it catalyzes the complete process. Number B, it provides the genetic blueprint for the protein. Number C, it creates the genetic code for to a specific amino acid. And number D, it modifies the messenger DNA molecules prior to the protein synthesis. Okay, let me check the chat box. Okay, so I have received a couple of answers, and all of you thinks that number B is the correct answer. Okay, let me check if number B is the correct answer or not. So the correct answer will be yes, very good. It is number B. It provides the genetic blueprint for the protein. Okay, so mRNA or the messenger RNA. So what does this messenger RNA do? So basically, so first the DNA is transcribed into the mRNA. Okay, so first the DNA, it has a genetic code which is transcribed into the mRNA. This mRNA then translated into the amino acids. Okay, so mRNA has a thing which is known as the codon, which is the three nitrogenous base together. It forms a particular codon, and each codon codes for a particular amino acid, and via this particular amino acids are joined together and form a particular polypeptide chain. Okay, so basically it is known as the central dogma of the molecular biology. For the DNA, it provides the particular genetic code. Okay, so how does it happen? So how DNA directs the protein synthesis? Let us first understand that. Okay, so first this is the double helix of the DNA, which we have seen in our last slide itself. So as you can see, in the inside of this helix, we have this one purine base is bonded with another pyrimidine base, right? So first, the strands of DNA unzip, okay, like this. This double helix it unzips and allow the free RNA mononucleotide to link separated with the strand. So as you can see here, first it is unzipped and then free RNA nucleotide linked to join to the separated strands. Okay, so after that this free RNA nucleotide approaches the unzipped DNA molecule to pair with the base to the DNA nucleotide. For example, if there is a thymine, adenine nucleotide or adenine RNA nucleotide will be coming and pairing up with this thymine. Okay, so like that it happens. This mRNA move the, so after this is the mRNA which is forming, right? So this is the formation of the mRNA. How the mRNA is formed from the DNA? So as you can see, first we have DNA. DNA is getting unzipped, RNA nucleotides are coming and forming the mRNA or messenger RNA. This messenger RNA moves from the cell nucleus to the ribosome. So this is the ribosome, which is the very important organelle for the protein synthesis. Okay, the mRNA, the which is formed from this unzipped DNA, it moves from the cell nucleus to the ribosome in the cytoplasm. It acts as a pattern or blueprint on which the amino acid is transported by the transfer RNA or tRNA. Okay, so on this mRNA, each code of this particular two nitrogenous bases creates a code, and each code is known as codon, which codes for an amino acid. So now the formation of protein on ribosome, the growing so each by understanding by reading the codons, the particular tRNA brings the amino acids. Okay, and this amino acids are attached with each other with the peptide bond, and polypeptide chain keep rising. So that's how this particular protein synthesis happens. So what is the role of this mRNA? So mRNA acts as a blueprint, or it acts as a pattern by which this particular protein synthesis happens. Okay, and how this mRNA forms? So this mRNA formed from a DNA. First the DNA it gets unzipped and free RNA nucleotide approached to the DNA to pair up with a particular base, and then it creates the mRNA. Okay, so that is how this particular protein synthesis happens. So yes, number B was the correct answer. Now let me go to the next problem. So problem number 3 says the position where the tRNA will bind to ribosome depends on its number A codon sequence, number B intramolecular hydrogen bonding, number C anticodon sequence, and number D hairpin like structure. Which of the following will be the correct answer for problem number 3? The position where tRNA will bind with ribosome depends on its which structure? Number A codon sequence, number B intramolecular hydrogen bonding, number C anticodon sequence, and number D hairpin like structure. Let me know A, B, C or D, which of the following you think is the correct answer for problem number 3. I will wait for a minute, then I will check the chat box. Meanwhile, let me know what do you think. What is the position where the tRNA will bind to ribosome? And the position, it depends on which structure or which particular sequence it depends on. Number A codon sequence, number B intramolecular hydrogen bonding, number C anticodon sequence, and number D hairpin like structure. So what will be the correct answer for problem number 3? Let me check the chat box. Okay, number D, number A, number C, number A. So I have got different answer. I have got 2 number A, 1 number C, and 1 number D. Okay, let me check which of the following will be the correct answer for problem number 3. So the correct answer will be, it is number C. Okay, anticodon sequence. The position where the tRNA will bind to the ribosome depend on the tRNA's anticodon sequence. Okay, now let us understand. So this is the structure of the tRNA. So as you can see, this is the structure of the tRNA. We have this nitrogen base lined up with each other. We have this D loop. Okay, then we have this sequence which is known as the anticodon stem, and this is known as the wobble position, and this is the anticodon sequence. Then we have this anticodon loop. We have a variable loop, and then we have a T loop, and then we have a acceptor stem, which is the 3' OH is here, which is the position where the amino acid is attached. The structure of tRNA, if you can remember I mean, try to practice the structure. Okay, just I mean, this particular amine, this guanine bases, they you try to remember. If it is not possible to remember, skip this, but remember these things, that this is the 5 prime, this is the 3' OH, this is the amino acid attachment site, and all the names of the positions. Okay, so the transfer RNA or tRNA is the crucial adapter unit which links the triplet code of the mRNA to specific amino acid. This means there has to be a different tRNA for each amino acid, and all the tRNAs are the clover leaf in shape and two different binding regions, opposite ends of the molecule. One binding region is for the amino acid, this one, and for the specific amino acid is covalently linked to the tertiary terminal adenosine residue. The other is a set of the 3 nucleic acid bases, which is the base pair of the complementary triplet of the mRNA molecule. A tRNA is having a particular anticodon will always have the same amino acid attached to it. Okay. So now how this thing happens? So this is the mRNA. Okay, so codon, which we have learned earlier, that it is a I mean, this nitrogenous base code. So okay, so there are several codons, and each codon is specific for a particular amino acid. Okay, when it is on the mRNA, it is known as the codon. When this particular sequence is on the tRNA, it is known as the anticodon. Okay, codon and anticodon with both are the complementary to each other. Now I will be showing how it happens. Okay, so let's see, these are the various these are the different parts for a protein synthesis. This we have, first we have this mRNA, this one. On mRNA we have this codons. Okay, so this, for example, AUG, it is a start codon. It is always known as start codon, and it codes for the amino acid methionine. Okay, so each and every amino acid we have a several codons. We have stop codons also. Okay, and this stop codons, they code when there is a stop codon, this particular synthesis of a chain will be stopping. Okay, and this polypeptide chain is released. So we have 3 stop codon, one UGA, one UAG, and one UAA. Okay, okay. So then we have this tRNA with a particular amino acid based on the anticodon sequence. We have this ribosomal units, this large and small. First is the initiation step. So there are 3 steps for the protein synthesis. One is the initiation, then elongation, and then termination. What is the initiation step? The translational complex form, and how this translational complex form, by bringing together all this mRNA, tRNA, large and small ribosomal subunits all together comes to form a translational complex, and the tRNA, it brings the first amino acid in the polypeptide chain to bind to the start codon. Okay, so that's how tRNA brings the particular amino acid. So now, as you can see, these are the complementary. As I have earlier mentioned, adenine binds with uracil in case of RNA. Okay, and guanine binds with cytosine and vice versa. So as you can see, if the start codon is AUG, the anticodon sequence will be U, A, and C. Okay, so based on that, this tRNA brings the amino acids. Okay, and this codon anticodon matches, and then the amino acids are there. So now, elongation step, tRNAs will keep bringing one by one amino acid based on other codons which are situated on this mRNA. Okay, and the group polypeptide chain will be forming by the peptide bond formation between the amino acids. Now after the completing the polypeptide chain, what happened? The release factor, there is another factor here which is known as a release factor. This release factor detects the stop codon, which is UGA, UAA, or UAG. Okay, these 3 are the stop codons. Then the stop codons, what they will do, the stop codons will be released. Okay, and it will the complete polypeptide chain, it will be released. Okay, and all the components of this translational complex, like the ribosome, mRNA, tRNA, all will be recycled for the next cycle of the polypeptide synthesis. Okay, so that's how this particular thing happens. Okay, so try to practice when I will be sharing this particular PPT. Read this thing carefully and try to practice. In these cases, the figures are also important. Okay, try to draw the figures by yourself. It is not a very difficult figure, but if you practice for 1 or 2 times, you can easily draw these figures. Okay, so these are important for the descriptive questions. Okay, which may be asked in your final examination. So it is better to start practicing from now itself. Okay. So yeah, number C was the correct answer for problem number 3. I will be now moving toward the next problem. So problem number 4, it says that in prokaryotes, the first amino acid in the polypeptide chain is, your options are number A methionine, number B N-formyl methionine, number B N-methyl methionine, number C N-formyl methionine, and number D all of these. In prokaryotes, the first amino acid in the growing polypeptide chain will be number A methionine, number B N-methyl methionine, number C N-formyl methionine, number D all of these. I will wait for a minute or two, then I will be checking the chat box. Okay. Which of the following is the first amino acid in the polypeptide chain in prokaryotes? In prokaryotes, the first amino acid in polypeptide chain is methionine and methyl methionine and formyl methionine, or all of these. Let me check the chat box. Let me see if there is any answer for problem number 4. Okay, I have got several answers. I have got 3 number C and 1 number D. Okay, let me check which of the following is the correct answer, number C or number D. Okay, so the correct answer will be, yes, it is number C is the correct answer. N-formyl methionine is the first amino acid in the polypeptide chain in prokaryotes. Okay, so this is the structure of the N-formyl methionine. It is only the formylation happens on this alpha NH2 of this particular methionine. So in case of the eukaryotes, methionine is the amino acid which is coded by the codon AUG, and it will be always the first amino acid in any polypeptide chain. Okay, so similarly in prokaryotes, N-formyl methionine does this particular job. So N-formyl methionine or F-met is modified form of the amino acid methionine, specifically where a formyl group is attached to the methionine amino group. It plays a crucial role in initiation of the protein synthesis in prokaryotes like bacteria and archaea, and the organelles of the eukaryotic cell like mitochondria and cytoplast. Protein synthesis initiation in prokaryotes. The protein synthesis begins with the for N-formyl methionine, which is carried to the ribosome by a specific transfer RNA or tRNA called the initiator tRNA F-met. Okay, the AUG codon, which signals to start of the protein synthesis, recognizes the F-met and T-F-met, tRNA F-met. This progress is facilitated by the initiation factors and requires energy. The specific sequence of mRNA, the Shine-Dalgarno sequence, helps to align the AUG codon with the ribosome's P site. Post-translation modification. After translation, the formyl group of methionine is removed by the deformylase enzyme. The methionine residue itself might also be removed by the aminopeptidases. This modification results in the mature protein from a non-formulated N-terminal methionine or another amino acid if methionine was clipped off. Significance. This N-formyl methionine acts as a signal marking the protein synthesized in the prokaryotes and organelles, differentiating them from the those made in the eukaryotic cytoplasm. In some cases, this N-formyl methionine can be involved in the protein degradation pathways, acting as the signal for the protein turnover. The presence or absence of this N-formyl methionine modifications can influence the stability and the function of the proteins. Comparison with eukaryotes, the eukaryotic cells, the initiator tRNA carries unmodified methionine for the protein synthesis, not this T-formyl N-formyl methionine. Eukaryotic proteins can undergo in terminal acylation, which is similar modification to formylation but with the acyl group instead of a formyl group. N-terminal acylation in eukaryotes can also be involved in the protein degradation pathways and act as a signal for targeting proteins to the proteasome or autophagy. In summary, this N-formyl methionine is a critical component in the initiation of the protein synthesis in prokaryotes and organelles, and its presence and absence can have significant implication for the protein maturation and degradation. Okay. Now I will be going to the next problem. Problem number 5 says, which of the following enzymes is crucial for the tertiary structure of DNA? Your options are number A topoisomerase, number B helicase, number C DNA polymerase, and number D ligase. Which of the following enzymes is crucial for the tertiary structure of DNA? Topoisomerase, helicase, DNA polymerase, or ligase? Again, I will be waiting for a minute, then I will check the chat box. Okay, let me know A, B, C, or D, which of the following you think is the correct answer of problem number 5. I am saying it repeatedly, even if your answer is wrong, it's completely fine. Okay, but at least try to answer these questions. Otherwise, I won't understand if you are following the class or not. Which of the following enzyme is crucial for the tertiary structure of DNA? Topoisomerase, helicase, DNA polymerase, or ligase? Which of the following enzyme is crucial for the tertiary structure of DNA? Let me check the chat box. So as I have mentioned earlier also, like protein in the DNAs or RNAs, we also have this primary, tertiary, secondary structures, okay, and quaternary structures also. So I have received 4 answers and all are saying that number A is the correct answer for problem number 5. Okay, let me check if it is correct, if it is true or not. So the enzyme that is crucial for the tertiary structure of DNA will be, it is number A, okay, topoisomerase, very good. So now let us understand regarding the topoisomerase, how the topoisomerase works, okay. Okay, I will be showing the figure. Okay, let's skip this figure together. So, the tertiary structure of DNA. The tertiary structure of DNA is often neglected or ignored, but it is important to the action of the quinoline group of the antibacterial agents and several anti-cancer agents. All these we will be discussing later in our course, okay. DNA is an extremely long molecule, so long, in fact, it would not fit into the nucleus if it is existed as a linear molecule. It has to be coiled in a more compact three-dimensional shape which can fit into the nucleus, a process known as the supercoiling. This process requires the action of the family of enzymes called topoisomerases, which can catalyze the seemingly impossible act of passing one stretch of DNA helix to another stretch. They do this by the temporarily cleaving one or both strands of DNA helix to create a temporary gap, then the resealing the strands once the crossover has taken place. Supercoiling allows the efficient storage of the DNA, but the DNA has to be uncoiled again if the replication and transcription takes place. If this uncoiling did not take place, the unwinding process catalyzed by the helicase enzyme that takes place during the replication and transcription would lead to increased tension due to the increased supercoiling of the remaining DNA double helix. You can demonstrate this by principle by pulling apart two strands of the rope or the sisal. The same topoisomerase enzymes are responsible for catalyzing the uncoiling process. So inhibition of this enzyme would effectively block the transcription and translation, okay, replication. So let's say we have a double helix of DNA. So you can try it if you have a rope which are intertwined with each other, which looks like the double helix of the DNA, and if you try to pull apart those two strands with opposite direction, you will be finding that there is a tension which is generated at as you are uncoiling that particular strands by pulling the rope. So you will be finding there is a tension. So this same type of tension generates in case of the DNA, and tension is relaxed by this topoisomerase enzyme. Okay, so these enzymes are very crucial in cases when the DNA is needed to be uncoiled, like in case of translation, replication, and so on. Okay, so so when there is a agents which block the action of topoisomerase, the inhibitors of the topoisomerase, they act as they also inhibit or block the transcription and translation process. So these agents are very crucial, especially in the anti-cancer drug discovery, also in the antibacterial drug discovery, the things which we will be discussing in detail later. Okay. So now first let us see how this thing works. Okay, let us see the in case of the figure. So topoisomerase, there are two to three types of topoisomerases. Okay, so majorly we will be seeing about this. There is a topoisomerase 1 and 2. So the topoisomerase 2 is a mammalian enzyme that is crucial for the effective replication of DNA. The enzyme binds to the part of DNA where two regions of the double helix in the near proximity. So as you can see, there is a crossover is here. Okay, and this crossover is generated when there is a supercoiling. So let's say we have this topoisomerase 2. So topoisomerase 2, what does it do? So it cuts on the double helix of the DNA in the two positions. Okay, as you can see here, and it creates a gap in this through this gap, this another. So as you can see, this is one strand, and this is the blue one is the another strand. This topoisomerase 2 cuts on this black DNA, and it creates a gap through which this blue strand of DNA can pass, and then resealing happens. Okay, which and then topoisomerase this enzyme it leaves. Okay, so that's how this particular thing happened. The enzyme binds to one of these DNA double helixes, the tyrosine residues. Okay, the tyrosine residues are used to nick both the strands of DNA. This results in a temporary covalent bond between the enzyme and resulting 5' end to each strand, thus stabilizing the DNA. The strands are now pulled to the opposite directions to form a gap through which the intact DNA region can be passed. The enzyme then reseals the strands and departs. Topoisomerase 1 is similar to topoisomerase 2 in that it releases the torsional stress of supercoiling DNA during replication, transcription, and repair of the DNA. The difference is that it cleaves only one strand of DNA, whereas topoisomerase 2 cleaves the both strand. The enzyme catalyzes the reversible transesterification reaction similar to so how this happens. So basically, this is here it is shown that it is cutting the DNA strand. How it happens exactly? So this topoisomerase have this tyrosine residue. Okay, and this is in the DNA we have this phosphoester bond, right? This tyrosine residue which has a phenolic OH, it acts as a nucleophile, attacks on the phosphate, and that's how this cleavage happens. Okay, so this is the mechanism by which the topoisomerase 2 splits the DNA chain. Okay, and this is the similarly it so topoisomerase 1 creates a cleavage complex with the single strand breaks. Relaxation of the torsional strands takes place by either by allowing the intact strand to pass through the nick or by free rotation of the DNA about the uncleaved strand. Once the torsional strand has been relieved, the enzyme rejoins the cleaved strands of DNA. Topoisomerase 4 is a bacterial enzyme that carries out the same process in the mammalian enzyme of topoisomerase 2 and is an important target for the fluoroquinolone class of antibacterial drug. These things we will be discussing when we have this topic of the antibacterial drug discovery. That time we will be discussing it. You do not need to memorize all these things, just read thoroughly and try to understand how it is happening, and these figures are important. Try to practice these figures and try to if you do not understand anything, if there is any doubt regarding this part, you can let me know via email, or also you can let me know in the next class, and I will be repeating the things. Okay, now let me go to the next problem. Problem number 6 says, which of the following statements regarding ribosome is true? Your options are: number A, ribosome of prokaryotes is larger than the eukaryotes. Number B, ribosome is made up of messenger RNAs and proteins. Number C, the eukaryotic ribosome contains 50S and 30S subunits. Number D, the prokaryotic ribosome contains a 50S and 30S subunits. Which of the following statements regarding ribosome is true? I will be waiting for a minute, then I will be checking the chat box. Okay. Try to figure out which of the following statement regarding ribosome is true. Read all the options carefully and then let me know. Which of the following statements regarding ribosome is true? This is very easy. I know all of you can answer this correctly. So let me check the chat box. Okay, I got four answers, and all the four are saying that, okay, five answers, and all the five are saying that number D is the correct answer. Okay, let me check if number D is the correct answer or not. Yes, number D is the correct answer. The statement which is true regarding the ribosome, that the prokaryotic ribosome contain a 50S and 30S subunit. Earlier, I have mentioned that ribosomes are very crucial organelle for the protein synthesis. Okay, ribosomes are small, rounded, dense particles of the ribonucleic protein. They occur either freely in the cytoplasm and the matrix of the mitochondria and chloroplast, or remain attached to the membranes of the endoplasmic reticulum and nucleus. They are the site of the protein synthesis in both prokaryotic and eukaryotic cells. The cells in which the active protein synthesis takes place, like pancreatic cells, hepatic parenchymal cells, plasma cells, thyroid cells, the ribosomes remain attached with the membranes of RDR. Okay, then these cells which synthesize specific proteins for the intracellular utilization and storage, like erythroblast skin, and contain large number of free ribosomes in cytoplasm. Ribosomes are usually designated according to the creating rates of the sedimentation. The sedimentation of the coefficient are expressed in the Sedgwick unit or S unit. Okay, this S is related to the size and molecular weight of the ribosomal particle. So in the prokaryotic and eukaryote, this ribosomes has different shapes. Okay, this prokaryotic ribosome, they contain a 50S and 30S subunit, and in total it is known as a 70S ribosome. The eukaryotic ribosome, which is known as the 80S ribosome, contains two subunit. One has the 60S subunit and one 40S subunit. Okay, so the statement which so in case of the statement number A, which says ribosome of prokaryote, it's larger than the ribosome. One second. Of eukaryotes, it is not true. Number B, which says ribosome is made up of the messenger RNA and proteins, it is also untrue. Number C, it says eukaryotic ribosome contain a 50S and 30S subunit, it is also untrue. But the last option, which says that prokaryotic ribosome contains a 50 and 30S subunits, it is true. Okay. So now let me go to the next problem. So problem number 7. Now we are moving toward your enzyme inhibition and the kinetics of the enzyme inhibition. Okay. So problem number 7 says, which of the following diagrams best describes the equilibrium associated with a competitive inhibition? Okay, where E represents the enzyme, S represents substrate, P represents the product, and I represents the inhibitor. Your options number A is this one, option number B this one, option number C is this one, and option number D. So option number E, it says that enzyme plus substrate, which forms the enzyme substrate complex, which forms the enzyme plus product. Okay, this enzyme is with inhibitor forms the enzyme inhibitor complex, so which forms the enzyme inhibitor substrate complex, which is also in equilibrium with this. This is the option number A. Option number B, enzyme and substrate form a enzyme substrate complex with which the inhibitor binds and forms the enzyme substrate inhibition complex. Number C, enzyme substrate, it forms the enzyme substrate complex, enzyme and inhibitor forms the enzyme inhibitor complex, and rest as follows. And number D is this one, where the enzyme and substrate inhibitor are interchangeable, and this is how this things happens. Okay. Now, let me know which of the following diagram best describes the equilibrium associated with the competitive inhibitor. There are different types or modes of enzyme inhibition. One by one, we will discuss all of those. So, to start with, I have chosen this competitive inhibitor. Now, let me know which of the following you think is the correct equilibrium associated with the competitive inhibitor. Let me check the chat box. Okay. Let me see if there is any answer for problem number 7. Okay. I got again 4 answers and all are thinking that number C is the correct answer. Let me check if number C is the correct answer for problem number 7 or not. So the correct answer, the correct diagram which represents the equilibrium in case of this competitive inhibitor will be very good. It is number C. Okay. So basically, competitive inhibition of enzymes. So the competitive inhibitor has roughly the same shape as the substrate, so it can fit into the active site. Okay, competitive inhibitor is able to fit at the active site when it is here the substrate is unable to bind to the enzyme and the desired reaction does not occur. The inhibitor and the substrate both compete with each other for the active site. The competitive inhibitor is often the product of the enzyme catalyzed reaction. When a lot of product is made, the product would compete with the substrate for the enzyme's active site, showing the reaction down. So as you can see here in the figure, this is the competitive inhibitor, this is the substrate. Both has similar, roughly the same shape. Okay, so the competitive inhibitor, it has similar shape to the substrate. That is why it competes with the substrate to bind to the active site. So now look at the equilibrium. We have enzyme and substrate, which forms the enzyme substrate complex. Enzyme inhibitor also can form the enzyme inhibitor complex because both have the same size and shape. From the enzyme substrate complex, we have this product formation, but not from the enzyme inhibitor complex. Okay, the normal enzyme maximum rate of the reaction can be reached when when we know how this happen. Competitive inhibitor, when the competitive inhibitor competes for the active site with the substrate. Now, with when the substrate concentration exceed the inhibitor concentration, the reaction will proceed and maximum rate will be achieved. Non-competitive inhibitors also are there. Those inhibitors don't compete for the active site. They attach to an alternative site of the enzyme, changing the shape of the active site, preventing the substrate from the binding. Non-competitive inhibition cannot be overcome by increasing the substrate concentration. Therefore, maximum rate of reaction will not be achieved regardless of the substrate concentration. This inhibition lowers the amount of the usable enzymes. Okay, so this is very important thing to remember, that since the competitive inhibitor and substrate both have roughly the same structure. Okay, so what happens basically, this competitive inhibitor, they compete for the active site, but if the substrate concentration we keep increasing the substrate concentration, the amount of the inhibitor molecule which can attach to active site will be lower. Okay, so at a time, so this competitive inhibition can be overcome by increasing the substrate concentration. Okay, since if there is a more number, let's say we have 100 molecule 100 enzyme molecules, so 100 active sites are there. Let's say we have around 50 inhibitor molecule but 1000 substrate molecule. So if the substrate molecule is so high, it will be definitely will able to fit the I mean go to the enzyme particular active site, replacing the inhibitor. So the competitive inhibition can be overcome by increasing the substrate concentration, but it is not the same, the case is not the same with the non-competitive inhibitors, because in case of non-competitive inhibitors, they bind to an alternative site, okay, rather, or to the some allosteric sites, and then change the active site. So now increasing the substrate concentration will not overcome this particular kind of inhibition. So this is the differences between this enzyme inhibitions, competitive and non-competitive. Okay, So now let me go to the next problem. Problem number 8 says, what is the mechanism of action of the cyanide poisons? Your options are number A, competitive inhibition, number B, non-competitive inhibition, number C, uncompetitive inhibition, number D, irreversible inhibition. What is the mechanism of actions of the cyanide poisons? Competitive inhibition, non-competitive inhibition, uncompetitive inhibition, or irreversible inhibition? Again, I will be waiting for a minute, then I will check the chat box. Okay. Let me know A, B, C, or D, which of the following is the correct mechanism of action for cyanide poisons? What is the mechanism of action for the cyanide poisons? Let me check the chat box now. Let's see how many answers I have received. Okay, number D, number C, number D, number D. So I have received number D and number C as two different options for this particular question. Let me check which of the following will be the correct answer. Okay, I have received three number D and one number C. Okay, let me check. So the correct mechanism of action for cyanide poisons will be, it is number B, non-competitive inhibition. Okay. Now, let us understand what are the non-competitive inhibitions. So, this is the steady state equation, or this diagram represents this particular kinetics of this non-competitive inhibitors. So, non-competitive inhibitor does not have structural similarity with the substrate, but it binds with the both free enzyme and enzyme substrate complex. This thing which you have to remember. Okay. So, non-competitive inhibitor, what is the speciality about this non-competitive inhibitor? Non-competitive inhibitor does not have any structural similarity with the substrate like the competitive inhibitors, and it can bind both with the free enzyme and also with the enzyme substrate complex. The simplified equation of the steady state is this one. As you can see, enzyme with enzyme inhibitor can bind to form enzyme inhibitor complex. With enzyme substrate complex also, it can bind to form the enzyme substrate inhibitor complex. The presence of the substrate has no influence on the ability of the non-competitive inhibitor to bind an enzyme or vice versa. Its binding alters the conformation of the enzyme and reduces its catalytic activity due to changes in the nature of the catalytic groups at the active site. Enzyme inhibitor and enzyme substrate inhibitor complexes are non-productive. So this is important to remember. Okay, this enzyme inhibitor complex and this enzyme substrate inhibitor complex, both are the non-productive. On increasing the substrate concentration to a saturating concentration does not reverse this inhibition, leading to an unaltered Km but reduced Vmax. Okay, so in this case, the Km remains unaltered in case of this non-competitive inhibition, but the Vmax is reduced. Okay, the maximum rate of the reaction cannot be achieved, since increasing a substrate concentration does not increase this rate of the reaction. Since it does not remove increasing the substrate concentration, like in last case, in the case of the competitive inhibitor, by increasing the substrate concentration, we can achieve the highest Vmax, but in case of non-competitive, it is not the same. Reversal of this inhibition requires special treatment like dialysis or pH adjustment. Okay, so these special treatments only can reverse this particular kind of inhibition. So example of non-competitive inhibition are mostly the poison. Cyanide and azide inhibit the enzymes with iron and copper as the component of the active site or the prosthetic group. For example, cytochrome C oxidase. Cyanides bind with the iron 3 of the in the heme of the cytochrome A3 component of the cytochrome C oxidase and prevents the electron transport to oxygen. Mitochondrial respiration and energy production cease and the cell death rapidly occurs in the central nervous system in the primary target for the cyanide toxicity. Acute inhalation of the high concentration of the cyanide like smoke inhalation during the fire and automobile exhaust provokes the brief central nervous system stimulation rapidly followed by the convulsion, coma and death. Acute exposure to the lower amounts to cause lightheadedness, breathlessness, dizziness, numbness and headaches. Cyanide is present in the air as hydrogen cyanide or HCN, in soil and water as cyanide salt, and in food like cyanoglycosides. Okay, so the cyanide poisons are example of the non-competitive inhibition. Now let me go to the next problem. Problem number 9 says which of the following inhibition is called the mechanism based enzyme inhibition. Your options are number A competitive inhibition, number B non-competitive inhibition, number C suicide inhibition and number D active site directed irreversible inhibition. Which of the following inhibition is known as the mechanism based enzyme inhibition? Competitive, non-competitive, suicide or active site directed irreversible inhibition. Again, I am waiting for a minute or two, then I will be checking the chat box. Okay. Let me know A, B, C, or D, which of the following you think correct answer. Or problem number 9, which of the following inhibition you think is called as a mechanism based enzyme inhibition. Which of the following inhibition is called mechanism based enzyme inhibition? Competitive, non-competitive, suicide, or active site directed irreversible inhibition. Let me check the chat box. Okay. I have received many answers. Number C, number C, number C. So all answers which I have received are saying that number C is the correct answer. Okay, let me check if number C is actually the correct answer or is there any different answer. So the correct answer will be yes, number C, suicide inhibition is known as the mechanism based enzyme inhibition. Okay, so what happened is, so in case of the suicide inhibition, a suicide inhibitor initially inactive attached to the active site of the enzyme, okay, then undergoes catalysis to form a product which occupies the active site and covalently binds to it. This binding alters the enzyme in such a way that the enzyme is now damaged irreversibly. So as you can see, suicide inhibitor, it looks like a substrate, fits to the active site perfectly, forms the enzyme inhibitor complex and undergoes the catalysis, and then the product which form a part of the product, it is attached with the enzyme product complex and it irreversibly, and now the enzyme is damaged and it cannot work anymore. So that's how we can say that this enzyme commits suicide by this mechanism, that is due to which it is known as a suicide inhibition. It is also known as the mechanism based inactivation. The inhibitor makes the use of the enzyme's own reaction mechanism mechanism to inactivate it. In suicide inhibition, the structural analog is converted to a more effective inhibitor with the help of the enzyme to be inhibited. For example, ornithine decarboxylase catalyzes the conversion of ornithine to putrescine, which necessary for the polyamine synthesis. When this ornithine decarboxylase in trypsinoma is inhibited multiplication of the parasite is arrested. Therefore, inhibitors of this ornithine decarboxylase enzyme such as the difluoromethane ornithine have been found to be effective against the trypanosomiasis or sleeping sickness. A similar mechanism is observed in case of the allopurinol, which is oxidized by the xanthine oxidase to alloxanthine, that is strong inhibitor of the xanthine oxidase. The anti-inflammatory action of aspirin also based on the suicide inhibition. Arachidonic acid is converted to the prostaglandin by the enzyme cyclooxygenase. Aspirin deacylates the serine residue in the active site of the cyclooxygenase. Thus, the prostaglandin synthesis is inhibited and so inflammation subsides. So, this is all about this suicide inhibition. We will be discussing about it later also. Okay. So, let me go to the problem number 10. Problem number 10 says, which of the following inhibitors occurs in the multimeric enzyme? Your options are number A, competitive inhibition, number B, non-competitive inhibition, number C, uncompetitive inhibition, and number D, irreversible inhibition. Which of the following inhibitions occurs in the multimeric enzymes? Again, I will be waiting for a minute and two, and then I will check the chat box. Let me know by then. A, B, C or D, which of the following you think is the correct answer for problem number 10? Which of the following inhibitions occur in the multimeric enzyme? Number A, competitive inhibition, number B, non-competitive inhibition, number C, uncompetitive inhibition, and number D, irreversible inhibition. So whenever you are reading about this enzyme inhibition, few things are very important. One is understanding how this inhibition is happening. And it is better if you can remember the steady state approximations. I will try to show you all the steady state approximation of each kind of enzyme inhibitions. Okay, and also the examples. So let's say you are given to write a short note on a particular enzyme inhibition. So these things which will help you to write the kind of notes. Okay. So let me know which of the following inhibitions occur in the multimeric enzyme: competitive, non-competitive, uncompetitive, or irreversible. Which of the following inhibitions occur in the multimeric enzyme? Let me check the chat box. Number B, number B, number B, number B. Okay, so all the answers which I have received are saying that number B is the correct answer. Let's see. So the correct answer will be, it is number C, the uncompetitive inhibition. Okay, so it happens in the multimeric enzymes. So now let us understand what is the uncompetitive inhibition. Uncompetitive inhibitor has no structural similarity with the substrate. It may bind to the free enzyme or enzyme substrate complex that exposes the inhibitor binding site. In binding, although so basically it binds with this enzyme substrate complex. Okay, so in binding, although away from the active site, causes the structural distortion of the active and allosteric site of the complex enzyme that inactivates the catalysis. This leads to a both decrease in the Km value and also in case of the Vmax value. This inhibition cannot be reversed by increasing the substrate toward the saturating concentration, and the reversal requires a special treatment like dialysis. This is similar to the non-competitive. This type of inhibition is rare and encountered in the multimeric multi-substrate enzyme, where the inhibitor competes with one substrate to which it has some structural similarity and is uncompetitive for the other. Okay, so this, remember, this happens in case of the, this is although rare, it encountered in the multi-substrate enzyme where the inhibitor competes with one substrate and has which it has some structural similarity and uncompetitive for other. The reaction without inhibitor will be like this. Okay, that in case of the in presence of the uncompetitive inhibitor, it acts as this. So this is the steady state inhibition like this. Uncompetitive reversible inhibition is rare, but it may occur in multimeric enzyme. Some example of uncompetitive reversible inhibitors are the inhibition of the key regulatory heme synthetic enzyme, your delta amino vulinate synthase and dehydrase, and the heme substrate by heavy metal ion like lead, and this heavy metals like lead from the mercaptides with SH thiol active site of the enzyme, and oxidizing agents like ferricyanide also oxidizes this SH to the disulfide linkage. The ability of the lithium to exert a profound and the selective psychopharmacological effect has been focused on the considerable research effort. There is increasing evidence that lithium exerts the therapeutic action by interfering with the polyphosphoinositide metabolism in the brain and prevention of the inositol recycling by an uncompetitive inhibition of the inositol monophosphate. Okay, few pesticides are uncompetitive inhibitor. Best known example is the herbicide N-phosphomethylglycine, or also known as the glyphosate or Roundup, an uncompetitive inhibitor. 3-phosphomethylcimate, 1-carboxyphenyl transferase. So these are the examples of the uncompetitive inhibitors. Okay. Now let me go to the next problem. Problem number 11. The drug that inhibits the attachment of tRNA to mRNA ribosome complex is number A ricin, number B tetracycline, number C streptomycin, and number D cyclohexylamine. Which drug inhibits the attachment of tRNA to the mRNA ribosome complex? Let me know A, B, C or D, which of the following you think is the correct answer. The drug that inhibits the attachment of the tRNA to the mRNA ribosome complex is ricin, tetracycline, streptomycin or cyclohexylamine. Which of the following is that drug that inhibits the attachment of tRNA to mRNA ribosome complex? Ricin, tetracycline, streptomycin or cyclohexylamine? Let me check the chat box. Let's see if there is any answer for this problem number 11. Okay, I am receiving answers. Number B, number B, number B. Number B is the, most of the people, you are thinking that number B is the correct answer. Let me check. So, the drug that inhibits the attachment of tRNA to the mRNA ribosome complex will be, yes, very good, it is number B, tetracycline. So this is protein synthesis inhibitions by different agents. Okay, we have this. So I know all of you know what is these things. This is the ribosome. This is the 50S and this is the 30S subunit. This is the prokaryotic ribosome. Okay, and we have this tRNA. These are the proteins. These are the amino acids which are brought by the tRNA to form this particular polypeptide chain. This is the protein synthesis site. This is your messenger RNA or mRNA. Okay, and this is the so first we have this is the direction for the ribosome movement. So this is the tetracycline which interfere with the attachment of tRNA to the mRNA ribosome complex. Okay, then we have chloramphenicol which binds with the 50S portion and inhibits the formation of the peptide bond. We have the streptomycin which changes the shape of the 30S portion, causing the code of the mRNA to be read incorrectly. So these are the different agents which inhibit the protein synthesis of the prokaryotes. Okay, tetracyclines basically what does they do? They block the ribosomal protein synthesis in bacteria. So this is, you can see there is a codon anticodon. This is the tRNA, and tetracycline it binds with the codon in this place. So the tRNA, so it binds with the this mRNA ribosome complex. Okay, and because it binds with this position, this tRNA cannot attach to this portion. So human mitochondrial ribosomes are 55S, 38, are 39S and 28. This inhibition of tetracycline is similar. Decrease cytochrome C synthesis, decrease OX-phos, and increase the aerobic glycolysis and lactic acids. Okay, so basically these are the this particular protein synthesis in bacteria protein synthesis. So tetracyclines are a very good class of the antibacterial agents because it inhibits the protein synthesis of the bacteria. Okay, this class we will be we will be learning in detail when we reach to our, I think, week 10 or 11, when there is a topic of the antibacterial drugs. For now, remember this thing that tetracycline inhibits the attachment of tRNA to the mRNA ribosome complex. Okay, let me go to the next problem. Problem number 12. It says that the drug that acts as a selective COX-2 enzyme inhibitor will be number A, valdecoxib, number C, rofecoxib, number C, celecoxib, and number D, all of the above. Which of the following drug acts as a selective COX-2 enzyme inhibitor? Valdecoxib, rofecoxib, celecoxib, or all of the above? Again, I will be waiting for a minute or two and then I will be checking the chat box. Okay. I am going a little quick because we have a couple of mathematical questions to solve in today's class, okay, which will take some time. So that's why I am going a little fast. But if you have any doubt or any query, if you want me to repeat any particular question again, please let me know. Okay, I will surely repeat that particular question. So problem number 12 says the drug that acts as a selective COX-2 inhibitor will be number A, valdecoxib, number B, rofecoxib, number C, celecoxib, and number D, all of the above. Which of the following is the correct answer for problem number 12? Which of the following drug acts as a selective COX-2 enzyme inhibitor? Valdecoxib, rofecoxib, celecoxib, or all of the above? Let me check the answers in the chat box for problem number 12. Okay, number D, number D, number D. All the answers which I have received are saying that number D is the correct answer, means all of the above are the selective COX-2 inhibitor. Let me check if it is correct or not. So the answer will be yes, number D, all of the above act as a co-selective COX-2 inhibitor. So these are the structures of the valdecoxib, rofecoxib, and celecoxib. Okay. So identification of isosomes that predominate in some tissues but not the others allows the possibility by designing the tissue selective enzyme inhibitors. For example, the non-steroidal anti-inflammatory drug or NSAID indomethacin. So this is the indomethacin. Okay. So this is used to treat the inflammatory disease such as the rheumatoid arthritis and works by inhibiting the enzyme cyclooxygenase. The enzyme is involved in the biosynthesis of prostaglandin, which acts as agents which are responsible for the pain and inflammation of the rheumatoid arthritis. Inhibiting this enzyme lowers the prostaglandin levels and alleviates the symptoms of the disease. However, the drug also inhibits the synthesis of the beneficial prostaglandins in the gastrointestinal tract and kidney. It has been discovered that the cyclooxygenase has two isozymes, COX-1 and COX-2. The both isozymes carry out the same reaction, but COX-1 is the isozyme that is active under the normal healthy condition. In rheumatoid arthritis, the normally dormant COX-2 becomes activated and produces excess inflammatory prostaglandin. Therefore, drugs such as valdecoxib, rofecoxib, celecoxib had been developed to be selective for the COX-2 isozyme, so it only production of the inflammatory prostaglandin is reduced. Selectivity is possible by taking advantage of the fact that the isoleucine group present in binding site of COX-1, whereas the corresponding group in COX-2 is valid. Rofecoxib was authorized in 1999, but had to be withdrawn in 2004 as it was linked to an increased risk of the heart attack and stroke when taken over the period of 18 months or so. Okay, so this is an example how the selectivity matters in medicinal chemistry. Okay, since COX-1 and COX-2 both are the similar isozymes, COX-1 is normal healthy condition, is it is it dominates, and COX-2 is not that much important in healthy condition. But in the cases like arthritis, the COX-2 becomes the activated, and so selective inhibitors are required to inhibit the COX-2, only not the COX-1, because inhibiting the COX-1 will reduce the beneficial prostaglandin, not the toxic ones. Okay, so that's how the selectivity matters in case of the inhibitor designs. Now I will go to the next problem. Problem number 13 says fetal hemoglobin has a higher binding activity for oxygen than the adult hemoglobin. In comparison to the adult hemoglobin dissociation curve, the fetal hemoglobin curve will be number A shifted to left, display a lower Km, number B shifted to right, display a lower Km, number C shifted to right and display a higher Km, and number D shifted to left and display a higher Km. The fetal hemoglobin has a higher binding activity for oxygen than the adult hemoglobin. In comparison to the adult oxyhemoglobin dissociation curve, the fetal oxyhemoglobin dissociation curve will be where it will be shifting to right or left, which show the lower Km or higher Km. Let me wait for a minute or two, then I will check the chat box. Try to answer the problem. Read the options very carefully. Try to draw the particular Michaelis-Menten curve. Okay. Think how it looks and then read the question carefully and let me know what will be the correct answer. Fetal hemoglobin has a higher binding activity for oxygen than the adult hemoglobin. So now how the curve should look like. Let me check the chat box. Let's see how many answers I have received for problem number 13. Number A, number A, number A. So I have received 3 answers and all are saying that number A is the correct answer for problem number 13. Okay. Now let me check if it is true or not. So the correct answer will be very good. The curve of the fetal oxyhemoglobin dissociation curve will be shifted to left and display a lower Km value. Okay, so this is the curve for the fetal hemoglobin, and this is the adult hemoglobin. So this is the partial pressure of oxygen. Okay, and this is, we have this saturation of the hemoglobin with oxygen. So as you can see, if we draw from the half maximal, if you draw perpendicular and then draw on this curves and then draw perpendicular on x-axis, you will see the Km value of this fetal hemoglobin dissociation curve is lower than the adult hemoglobin dissociation curve. Now, why is this so? The fetal hemoglobin has a higher affinity for oxygen at all partial pressures. Okay, this ensures that oxygen is transferred to the fetus from the maternal blood across the placenta. The partial pressure of the fetal tissues is very low due to the high metabolic rate associated with the fetal growth rates. Although the fetal hemoglobin has a higher affinity for oxygen, in such a low partial pressure of environment of the fetal tissue, it unflows the oxygen readily. At the birth, the fetal hemoglobin is replaced with the adult hemoglobin. So in the adult hemoglobin, we have two alpha and two beta chains. Okay. In the fetal hemoglobin, we have two alpha and two gamma chains. Okay, the polypeptide gamma chains are enabled to fetus to have a greater affinity to oxygen, and this ensure that enough oxygen is being delivered to the organs for a developing fetus. Okay, so this remember, this lift shifts to the left and raises to the lower K. Now I will go to the next problem. Problem number 14. It says, which of the following mechanism of action of the orlistat in non an anti-obesity drug? Your options are: number A, competitive inhibition; number B, non-competitive inhibition; number C, uncompetitive inhibition; and number D, irreversible inhibition. Which of the following is the correct mechanism of action for the orlistat and anti-obesity drug? Competitive inhibition, non-competitive, uncompetitive, or irreversible inhibition? Let me wait for again another minute, and then I will check the chat box. Meanwhile, let me know A, B, C, or D, which of the following you think is the correct answer for problem number 14. What is the mechanism of action of orlistat and anti-obesity drug? Competitive inhibition, non-competitive inhibition, uncompetitive inhibition, or irreversible inhibition? Let me check the chat box. See what is the correct answer. Okay, I have received two answers and both are saying number D is the correct one. Let us first check what is the correct answer and then let us discuss, okay, the mechanism of action of orlistat. So the correct answer is yes, it is number D, irreversible inhibition. Okay, so so first understand what does this irreversible inhibitors do. So irreversible enzyme inhibitors can form a covalent bond to the key amino acid with in the active site of the active site of the enzyme and permanently block to the affected enzyme. The most so this is what the irreversible inhibitors do. Okay, they form a covalent bond with the key amino acid in the active site of the active site of the enzyme, and then permanently damages the enzyme. The most effective irreversible inhibitor are those that contain an electrophilic functional group capable of reacting with the nucleophilic group present in the on the amino acid side chain. Invariably, the amino acid affected are either the serine or the cysteine, because these amino acids are often present in the active site and contain nucleophilic OH and SH group respectively. This is that are involved in the mechanism of action sorry, of the many enzyme catalyzed reaction. Electrophilic functional groups used in the Irreversible inhibitors include the alkyl halide, epoxide, alpha beta unsaturated ketone or strain lactone or lactams. The highly toxic nerve agents contain electrophilic fluorophosphonate groups and are irreversible inhibitor of the mammalian enzyme. Irreversible enzyme inhibitors are not competitive inhibitors. In increasing the concentration of the substrate will not reverse their inhibition, as the inhibitors cannot be displaced from the active site. This can cause problems if the buildup of the particular substrate leads to the toxic side effects. For example, the monoamine oxidase inhibitors block the metabolism of noradrenaline and have antidepressant activity. Unfortunately, the metabolisms of substrate other than the noradrenaline is also inhibited, leading to the buildup of this compound and the serious side effect. Okay, so now let us go to the orlistate. So this is the structure of orlistate. As you can see, it has a strained lactam ring okay. So fat in the diet is composed mainly of the triglycerides, which are digested in the small intestine to the fatty acid and to monoglycerides. The digestion products are absorbed and act as a building block of the fat biosynthesis of our body. The enzyme pancreatic lipase is responsible for catalyzing the digestion of fat, so inhibition of the enzyme will result in the reduced absorption of the glycerides and the fatty acids from the gut. Consequently, lesser fat will be synthesized in our body. Orlistat is the anti-obesity drug which acts as an irreversible inhibitor of the pancreatic lipase as a result of presence of electrophilic four-member lactone ring, and this acylates the serine residue in the active site, which is a part of the catalytic triad of serine, histidine, and aspartic acid. Okay, so here is the enzyme pancreatic lipase, which is responsible for the fat digestion in our body. It is irreversibly inhibited by this orlistat, which is an anti-obesity drug. This contain an electrophilic lactone ring, which is a four-member ester, cyclic ester. Okay, and the nucleophilic serine reacts with this orlistat and covalently binds this particular, and now this serine cannot act as electrophilic to the substrate. So this is how this particular inhibition occurs. Okay, now I will go to the next problem. Problem number 15 says inhibition of cytochrome P450 by tylenic acid is an example of dash inhibition. Your options are competitive, option A and option B non-competitive, option C suicide, and option D active site directed irreversible inhibition. The inhibition of cytochrome P450 by tylenic acid is an example of which kind of inhibition? Competitive, non-competitive, suicide, or active site directed irreversible inhibition. Inhibition of cytochrome P450 by tylenic acid is an example of which kind of inhibition? Competitive, non-competitive, suicide, or active site directed irreversible inhibition. So, what will be the correct answer for problem number 15? Let me check. Okay, number D, number C. So, I have received equal numbers of number C and number D as the answers. So, let me check which of the following will be the correct answer, number C or number D. So, the correct answer will be, it is number C, suicide inhibition. Okay. Earlier also I have mentioned this thing, the suicide substrate are the agents which are converted to highly reactive species when they undergo an enzyme catalyzed reaction. They form covalent bonds to the enzyme and inhibit it irreversibly. In some cases, they can cause toxicity. For example, the diuretic agent tylenic acid had to be withdrawn from the market because it was found to act as a suicide substrate for the cytochrome P450 enzyme and involved in the drug metabolism. Unfortunately, the metabolic reaction carried out by these enzymes and enzymes converted tylenic acid to the thiophene sulfoxide, which is proved to be highly electrophilic. This encouraged a Michaeli reaction leading to alkylation of the thiol group in the enzyme's active site. Loss of the water from the thiophene sulfoxide restored when the thiophene ring and resulted in formation of the covalent link to the enzyme, thus inhibit the enzyme completely. So this is the cytochrome P450. Okay, we have a thiol group here in the active site of this enzyme. This is the structure of the tylenic acid. So after oxidation in presence of the cytochrome P450, NADPH and oxygen, what happens? This is the suicide substrate which is formed. Okay, this suicide substrate is the sulfoxide. Basically, this is a sulfoxide which acts as an electrophile, and when reacting with this nucleophilic thiol, it forms this covalent linkage, which is which is this covalent linked enzyme, which cannot further perform any enzymatic reaction. Okay, so this is the example of this irreversible suicide inhibition by this in the active site of cytochrome P450 by tylenic acid, which acts as a suicide substrate. Okay, now let me go to the next slide. So the mechanism is easy. If so, there is nothing but an electron going withdrawing. So first, the oxidation creates the sulfoxide, which is a plus and negative charge here. Thiol lone pair attacks on this position, and then the double bond rotation happens like this. Okay, so here also, so this double bond rotation. So this is the aryl. Let's say so. This alkylation makes like this. Then the negative charge moves back here, and again double bond rotation. So makes there is nothing but a double bond and electron rotations. Okay, so if you practice it 1 or 2 times, I think you will understand the mechanism. Again, I am saying if you do not understand the mechanism, let me know in the next class. I will repeat it. Okay, but I think you will be understanding once I share the slide. If you do it by yourself 1 time, I think it will be clear to you. Now let me go to the next problem. Problem number 16 says which inhibition has the apparent Vmax value as the Vmax of the normal enzyme catalyzed reaction. Your options are number A competitive, number B non-competitive, number C uncompetitive, and number D mixed. Which inhibition has the apparent Vmax value same as the Vmax of normal enzyme catalyzed reaction? Competitive, non-competitive, uncompetitive, or mixed inhibition? Already I have mentioned this thing 1 time. So I know you can correctly answer this. Which inhibition has apparent Vmax value as the Vmax of normal enzyme catalyzed reaction? Which inhibition has the apparent Vmax value same as the Vmax of the normal enzyme catalyzed reaction? Competitive, non-competitive, uncompetitive, or mixed? A, B, C, or D, which of the following will be the correct answer? Let me check the chat box. I have mentioned already, I have mentioned it 1 time, so I think you can answer it. Number A, number A, number A. So I have received 3 answers and all are saying that number A is the correct answer. Okay, let me check. So the correct answer will be yes, very good. Number A, competitive inhibition. The apparent Vmax value is same as the Vmax of normal enzyme catalyzed reaction. So this is how the curve look like, Lineweaver bark plot looks like. So this thing I have already mentioned. Okay, this how this steady state approximation. So kinetically, the inhibitor binds with the free enzyme reversibly to form an enzyme inhibitor complex. Okay, so these are the the competitive inhibitor reduces the ability of the free enzyme for the substrate binding. The total enzyme E0 exists in the reaction maximum as the free enzyme and bound enzyme and EI. So okay, so E0 is the total enzyme equal to free enzyme E plus bound enzyme ES and EI. Okay, in presence of inhibitor, the Michaelis Menten constant Km is the normal reaction, is increased to the new Km is the this one. Okay, where the dissociation constant is Ki equal to concentration of E into concentration of I divided by Ei. So V0 equal to, now this is the Michaelis Menten equation, Vmax into S divided by this is the new Km. Okay, so new Km plus S. So now if you solve all this thing, you will find that the Vmax values are same, but the Km value has decreased. Okay, so this is the so the Km value. So however, so you do not need to memorize all these things. I have only given you for your understanding. Okay, but try to remember what is the value of the new Km, and the Vmax remains the same, so it does not matter. But try to remember what is the value of the Km. Okay, so now Km equal to 1. So this is the new Km. Km equal to normal Km into 1 plus I by Ki. Okay, where Ki is the E into I divided by Ei. So these are the values. So if you see the graph, you will see the graphs have different slope, but the intercept is the same. So in the Lineweaver bark plot, we know the intercept value gives us the Vmax value. Since the intercept value is same, so the Vmax will remain same, but the Km will be different. Okay, so the Km will be decreasing. So this is the so in the presence of the Michaelis, sorry, in the presence of the inhibitor, the Michaelis came of the normal reaction increased to new Km. Okay, so that was my mistake. Okay, so the Km value will increase and the Vmax will remain same. So this particular equation, you do not need to memorize this. Just I have given you for your understanding. So try to remember the values if possible. Okay, what is the value of this Ki? The dissociation constant, this is the value of the new Km. The remembering this two will work fine. Otherwise, you don't need to memorize other things. Let me go to the next problem. So the problem number 17 says, which inhibition study deals with the decrease of the value of both Vmax and Km with the increase of the inhibitor concentration? Your options are number A competitive, number B non-competitive, number C uncompetitive, and number D suicide. Which inhibition study deals with the decrease of both the value of Vmax and Km? In our last question, we have seen that in case of competitive inhibitor, the value of Vmax will remain same and the value of Km will be increasing, right? But which inhibition study you have to identify will deals with the decrease of both Vmax and Km? Competitive, non-competitive, uncompetitive, or suicide? Again, I will wait for a minute, then I will check the chat box. All these are associated with the Lineweaver bark plot, which we have discussed in our last class. So along with the Michaelis-Menten equation and Michaelis-Menten plot, the Lineweaver bark plot is very important, the 1 by V versus 1 by S plot. Okay, and there is another plot which is known as the Hans-Wolf plot, which we will be discussing in today's class itself. It is also similar to the Lineweaver bark plot, just a little change here and there. But the Lineweaver bark plot is most acceptable and most used form, because in case of normal Michaelis-Menten constant, it is not possible for us to tell when if the Vmax is reached or not. But in the Lineweaver bark plot, since it is the reciprocal plot, it is easier to identify the Vmax and Km values. Which inhibition study deals with the decrease of the value of both Vmax and Km with increase in the inhibitor concentration? Let me check the chat box. Number C, number C, number C. So I have received number C as the answer for this problem number 17. Let me check. Very good. Number C is the correct answer. Uncompetitive inhibition deals with the decrease of both VM and KMS. So this is very important. Okay. The Lineweaver-Burke plots for the enzyme inhibition, how the plots look like. So red one is the with inhibitor, blue one is without inhibitor. Okay, so in case of the competitive inhibition, I have already discussed that Vmax is unaffected and Km is increased. This is the particular plot. Okay, so in case of the uncompetitive inhibition, both the Km and Vmax is reduced. Okay, in case of the uncompetitive inhibition, as the this both values are reduced, and in case of the non-competitive inhibitor, the Km is affected. Okay, and in case and the Vmax is reduced. Okay, so as you can see in here, in case of this, so we know the slope of this Lineweaver-Burke plot is Km by Vmax, and the intercept is 1 by V. Okay, so in case of the uncompetitive inhibitor, as you can see with inhibitor, the value of 1 by the intercept is increased. As you can see, the intercept value is increased. So if 1 by Vmax value is increased, okay, so if 1 by Vmax, let's say in presence of inhibitor, it's greater than 1 by Vmax without inhibitor. Okay, so then we can say that Vmax is less than Vmax in presence of the, okay, sorry, just a minute. So then we can say that the Vmax value in presence of inhibitor, it is decreasing. Okay, so then normal, in case of the normal Vmax value, it is like this, Vmax in presence of the inhibitor. It is you can see it is from this graph. Okay, and the slope value 1 by km by Vmax. So if Vmax value is decreased and the slope remains same, then the km value must also reduce. Okay, so that is the graphs how it looks like. In case of the non-competitive, the things are the same. Okay, now here also the km remain unaffected. Okay, the Vmax is reduced. That is why the Vmax is reduced, and Km remains unaffected. Since Km remains unaffected and Vmax is reduced, the slope also reduces. Okay, so that's how the Lineweaver-Burke plots are different kind of inhibition looks like. Now let me go to the next problem. Problem number 18 says the turnover number for chymotrypsin is 100 per second, and for the DNA polymerase it is 15 per second. The velocities of the reaction catalyzed by the both enzyme at saturating substrate level could be made equal if dash times more DNA polymerase than chymotrypsin were used. Fill in the blanks. Your options are number A 1500, number B 0.15, number C 67, and number D 6.7. Again, I am repeating, the turnover number for chymotrypsin is 100 per second, and for DNA polymerase it is 15 per second. The velocities of the reaction catalyzed by both enzyme at saturated substrate concentration can be made equal if dash times more DNA polymerase enzyme is used than chymotrypsin. Now you have to identify you have to fill in the blanks. Your options are 1500, 0.15, 67, and 6.7. I will be giving you 2 minutes, okay, 2 to 3 minutes. Then I will check the chat box, and then I will show you how to solve this kind of question. Read the question very carefully. It is very easy. Just read the question carefully and then try to solve it. The turnover number for two enzymes are given, and if the velocities of the reaction was to be made same at the saturated level, what should be the concentration of DNA polymerase and chymotrypsin? Should be the ratio? Okay, let me check the chat box because I have to discuss this question in detail. So let me be quick in checking the answers. Okay, I have received only one answer which says that number D is the correct answer. Let me check if it is so. So the correct answer will be very good. Number D is the correct answer. Now let us understand what is the turnover number. So the turnover number is defined as the number of product molecules formed by each enzyme active site per second. Okay. So it is Vmax equal to Kcat into enzyme in total, concentration of total enzyme. Kcat is the frequency of catalysis, or it is the turnover number which has a unit of second inverse. Okay. The value of Kcat is the measure of the catalytic efficiency. Fastest enzyme have the highest Kcat values. Kcat is a true constant. Its value only depends on enzyme's catalytic power. Okay. So Vmax cannot be a true constant because it depends on the enzyme concentration. So the Michaelis-Menten equation we know is equal to V0 equal to K2 S0 E0 divided by S0 plus Km. Okay. At the high substrate concentration, when S0 is equal to is much greater than Km, it looks like this. The rate equal to K2 E0 equal to Vmax. Okay. So this Vmax is the rate attains the maximum value, and the reaction is a zero order with respect to the substrate concentration. But E0 is there. E0 is the total enzyme concentration. This K2 is the turnover number for the enzyme and represents the number of molecules converted to the product per unit time by one molecule of enzyme. So this you have to remember. This equation is very important. Okay. This Vmax equal to Kcat into the E total. E total is the total enzyme concentration. Vmax is the highest rate of the reaction, and Kcat is the turnover number of that particular enzyme. Okay. So now according to the question, let's assume that Xc is the strength or in molarity or the concentration of the chymotrypsin enzyme, and Xd is the concentration of the DNA polymerase enzyme. Now if this equation holds true, then we can say Vmax of chymotrypsin equal to this turnover number into the concentration of chymotrypsin. Chymotrypsin's turnover number is 100. So we can say Vmax of chymotrypsin equal to 100 into the Xc, which is the concentration of the chymotrypsin enzyme, and the Vmax of the DNA polymerase equals to 15 into the concentration of DNA polymerase, right? If the velocities or to both the Vmax values are same, then we can say 100 into Xc equal to 15 into Xd. Okay, now Xd by Xc equal to 100 by 15 equal to 6.7. Means the DNA polymerase enzyme has the concentration of DNA polymerase has to be 6.7 times more than that of the chymotrypsin to make both of the reactions Vmax value same. Okay. So this is how you have to solve this kind of question. Whenever the question comes regarding the turnover number, you have to solve it using this particular equation. Vmax equal to K2 into A0. K2 is the turnover number and E0 is the enzyme concentration, the total enzyme concentration. Okay. I hope it is clear to all. Do I need to repeat this question, or is it clear? Let me check if everybody has understand this question, or do I need to repeat this one? Okay, very good. Try to do it by yourself once I share the slide. Okay, and let's to check if you can if you are understanding this particular thing or not. If there is any issue, I will repeat it. Okay. Now let me go to the next slide. Problem number 19 said, consider the plot of the concentration of S by V0 versus S plot of the Hans-Wolf. The results obtained from the graphs are the intercept value is 0.25 and the slope is 0.0005 millimolar second inverse. What is the Km of the reaction? Your options are number A 5000 millimolar, number B 500 millimolar, number C 2500 millimolar, and number D 250 millimolar. So as I have mentioned, the Lineweaver-Burke plot, what is the Lineweaver-Burke plot? We do 1 by V0 versus 1 by S, right? So the Hans-Wolf plot is same. Just you have to multiply the Lineweaver-Burke equation with the substrate concentration. Then you will get the particular Hans-Wolf plot. Try to figure out how to do it. I have already given you the hint that you have to solve it with the help of the Lineweaver-Burke equation. Just you have to rearrange the Lineweaver-Burke equation to receive to such that you get the this versus this plot. And from that you can easily identify the intercept and slope, and if you put the values you will get the answer. I will give you 2 to 3 minutes, then I will check the chat box, okay? Try to do it, okay? Okay, we have now does not have much time left and I have to solve this, so I'm just checking the chat box, okay? I think I have received many answer. Number B is the answer as the answer I have received. Let me check if number B is the correct one or not. The correct answer will be very good. It is number B, 500 millimolar. So as I have already mentioned that you have just multiply, you have to just multiply that particular your Lineweaver-Burke equation with the substrate concentration to have the Hans-Wolff plot. So this is the Hans-Wolff equation, okay? And it is also can be rearranged from this this Michaelis-Menten equation. You can also get this. So you have to rearrange in such a way that you get this kind of form where it can be, I mean, where it can be compared with our linear equation of y equal to mx plus c, where your m value is this one, okay, c intercept value is this one. So here in case of Hans-Wolff plot, your intercept value is km by Vmax and your slope is 1 by Vmax. So according to the question, intercept value km by Vmax is 0.25 and 1 by Vmax, the slope value is given as 0.0005 millimolar inverse. So Vmax is 2000 millimolar and km is 0.25 into 2000 equal to 500 millimolar, okay? So this is very simple actually. Just you have to all the thing you can derive in case of this enzyme kinet. I mean these things from the Michaelis method. This is the basic equation, and from it you can derive both Lineweaver-Burke plot and also the Hans-Wolff plot. Okay. Now I will go to the last question of today's session, which says, which of the following statements regarding genetic polymorphism is false? Your options are: number A, personalized medicines are required to combat this disease. Number B, it occurs due to the mutation of the gene. Number C, the genome remapping of the patient is not required to cure the disease. And number D, this disease leads to the different individual having subtly different gene sequence. Which of the following statements regarding genetic polymorphism is false? Which of the following statements regarding genetic polymorphism is false? Number A, personalized medicines are required to combat this disease. Number B, it occurs due to mutation of gene. Number C, the genome mapping of the patient is not required to cure the disease. And number D, the disease leads to different individuals having subtly different gene sequence. I will wait for a minute, then I will check the chat box. Let me check the chat box. Okay, let's see. So number C is the most given answer. Let me check if number C is correct or not. So the correct answer will be yes, it is number C. Genome mapping of the patient is not required to cure the disease. Okay. So genetic polymorphism, naturally occurring variants of the gene structure that occur in more than 1% of population. Polymorphisms may influence the drug's action by changing its pharmacokinetics or its pharmacodynamics. It is also known as the intersubject variability. It paves the path for the individual rational dosage regimen. Differences are observed in the metabolism of the drug among the different races called the ethnic variations. Genetic polymorphism and personalized medicine, the process of replication is not 100% perfect, and occasionally the mutation can occur. If mutation does not prove fatal, it will be carried on from generation to generation. This leads to different individuals having subtly different gene sequences. On average, there is a difference in one base pair in every 1,000 base pairs between individuals. This is known as genetic polymorphism. As the nucleic acid bases act as a code for amino acids in protein, the difference at this level results in different amino acid being introduced into the protein, which may or may not have effect on protein's activity or function. Genetic polymorphism has important consequences with respect to the susceptibility of the individuals to disease, and also kinds of the disease therapies are best suited for individual. A detailed knowledge for patient's genome opens up the possibility to predictability and preventing the disease, as well as choosing the ideal drug therapy for the patients should be occurred. This is known as the personalized medicine. Okay. So the statement which is the number 3, which says genome mapping of the patient is not required, it is false. Okay, genome mapping is very important and it is required in case of the genetic polymorphism. So this was the last question of today's session. So today what we have discussed, we have learned about different enzyme inhibitions and the details, that their steady state approximations and so on. So now I will share my email ID. Okay, let me just first share it. So this is my email ID. If you have any doubt or any query, please let me know through this email before the next class so that I can discuss that particular query in the upcoming class. Okay, so today it was the week 3 of live session for our NPTEL course of the medicinal chemistry, and today we have learned about different enzyme inhibitions and their steady state approximations, and we also did some mathematical questions. Okay, so thank you all. If there is no doubt, I will be closing the session for today. We will be meeting on the next Monday with our week 4 course content and live session. Okay. Till now, whatever we have learned, if you have any doubt or any query from any previous class, please let me know via email. Okay. If there is no doubt, I will be closing the session for today. Thank you all for joining. Thank you all for participating in the question answer session. I will be sharing the slides and the class recording soon. By tonight or by tomorrow morning, it will be available on the SWAYAM portal. Okay, you can download it and you can read. Thank you all. I am closing the session.